to welcome all of you for the talk 3 of the Precision Oncology Asia webinar series. Many of you have been regular attendees of this particular webinar series. And for those who are new to this particular talk, just a brief introduction. I'm Hitesh Goswami. I'm co-founder of 4Base Care Genomics. We are a precision oncology company. And Precision Oncology Asia webinar series is our effort in terms of bringing together experts from the field of oncology research clinic side to come together and share their knowledge and experience in how precision oncology is being applied in clinics today. So it's my great honor to welcome today Dr. Amit Dutt and Dr. Kumar Prabhash. So Dr. Kumar Prabhash, with more than 2 decades of experience in treating cancer patients, he is professor and medical oncologist from Tata Memorial Hospital. He has been involved very actively in translational and clinical research, one of the few clinicians who gets actively involved in basic research also, and has more than 17 publications and multiple talks in ESCO and ESMO kind of conferences. And along with that, we have Dr. Amit Dutt, who is a Shanti Sarup Bhatnagar Award, one of the most highest honors in multidisciplinary science awards in India. And his lab at ACTREC TMC in Mumbai, they essentially work on identifying somatic mutations in human cancer cells and associating those mutations with different kind of treatment options. So I would not take much of everyone's time and welcome Dr. Amit Dutt and Dr. Kumar. And Dr. Amit Dutt will be starting the presentation, and I'll be taking the questions at the end of it. Welcome everyone again.
So I'm Amit Dutt from ACTREC, basic researcher. And today we are going to talk about, I'm going to give you a realm of a glimpse of the basic research that has been happening at my group here at ACTREC. And I will be restricting myself to just a few aspects of the work that what we have been doing. And from there on, we will be moving on to the clinical aspect of it that Dr. Kumar Prabhash will be looking at, how these work can be translated or how it has already been translated. So I would go a bit horizontal. What that means is that I would go across 5 different diseases to give a glimpse of what kind of approaches that we take here at ACTREC, that is the Tata Memorial Basic Research Center, and what kind of discoveries that what we make over here. But then I would like to emphasize that we are limited with these discoveries to bring it to the patients, had it not been the Tata Memorial Center, and had it not been working along with the clinician together right from the day one. That is the basic success of like any of these translation kind of like approaches. So my slide doesn't move. Yes. To give a brief perspective before I get started, this is how the cancer incidence is being projected across the globe. That is projected to change over the times to come. Over next 20 years or something on, you would see that there is going to be an increase in the cancer incidence that is across the globe. However, what is striking over here is that if you would see the distribution across the countries with a higher income group and the countries with a lower income group, you would see a stark difference. While there appears to be a kind of a plateau in the increase in the incidence of cancer in the countries with a higher income group, there doesn't seem to be a similar. In fact, there is a completely opposite effect that what we kind of get to see in countries of lower or middle income groups such as India and China. In fact, as per the recent reports suggest that it's not just a plateau that what we have is that what they are attaining in these countries. This year is far more promising. It looks like that their trend is more towards the downward as well. So they are already kind of like moving towards a downward trend. However, this is not the case in the countries with the middle or lower income groups such as India. And there are several reasons associated with this. I won't go into the details of it because that's not exactly the focus of the talk of today. But I'm going to tell you that cancer as a disease, what we get to see in these countries, particularly in India, to a certain extent is also distinct than what we get to see in the Western world. Uh, Whether it is the etiological agent, whether it is the ethnicity, we really don't know. Even when we would look at the incidences of cancer, here are the list of incidences of cancer among women, and on the right are the incidences of cancer among men. You would see that cancers such as cervical cancer in India, here it kind of ranks two among women. And if you would look at among men, oral cancer happens to be one of the most prevalent cancer in India. Now, that's where what I meant to say, that how it is different from what you get to see in the Western world. These two cancers are not major tumor types, not major diseases in the Western world, but they are more of a disease, and they are such of a higher incidence in this. And now to understand these diseases, or to find therapeutically relevant targets that will be translated, an initiative has to take place from this part of the world. And that's what primarily my laboratory has been focusing here at ACTREC in Navi Mumbai. So it's not just the incidence or the different kinds of tumor types that is different in the Indian population or Indian subcontinent, but also, which is very striking that I want to emphasize, is that this incidence, which has been increasing, if you would compare from 1990 to 2016, it has been great, has doubled per year. And what is more of a worrisome is the number of deaths. It has gone up from 0.3 million to 0.8 million. In overall, still the number of cancer incidences stays one-third to what we get to see in India as compared to the developed country. However, the number of patients who get cancer, they're more likely to die in the Indian subcontinent. So we definitely require a much more detailed understanding of what's so different underlies in these cancer genome. And that's where my laboratory at Tata Memorial kind of like comes into it. So I would start with, as what I said, I would go horizontal and would abruptly leave hop on from one cancer type to the other cancer type, because we can leave it for the discussion that how this can be translated. To begin with, I would start with lung cancer. This was one of the first most project that I started in the laboratory at when I joined my lab here, when I started my laboratory here at Tata Memorial Center, along with my friend Dr. Kumar Prabhash at the Tata Memorial Hospital. So we started with one of the major type of lung cancer, that is the non-small cell lung cancer. And among those, what I wanted to talk about today is one of the subtype, which is the most prevalent subtype among the lung cancer, and the discoveries and the advancements that what we have made, along with Dr. Kumar Prabhash, in adenocarcinoma. I would also touch base upon the squamous cell carcinoma, some recent discoveries that what we have found, and this is one comparatively much of the less studied cancer as compared to adenocarcinoma, possibly purely because of its sheer incidence. We have also characterized now the genome of the small cell carcinoma, but today I'm not going to talk about that. Among lung cancer, I'm going to talk about the adenocarcinomas and the squamous cell carcinoma. So in adenocarcinoma, what is already known is that targeted therapy has or personalized medicine has gone, which happens to be the basic focus of today's presentation. One of the major success, the tumor type where it has kind of like seen besides melanoma, is the lung cancer. So here is a CT scan of a lung cancer patient, and what you get to see over here is these are the nodules, and this patient happens to be positive for a molecular alteration, and this molecular alteration happens to be in a gene which is an enzyme called as EGFR, an epidermal growth factor receptor protein. Mutations in this EGFR, if a patient is harboring, now we know that if instead of giving generic chemotherapy to these patients, if they receive a targeted therapy, that is a small molecule inhibitor targeted against the EGFR, which are mutated, you get to see a remission in just 2 months. And this is a kind of a magical bullet, and that is very much into the clinical practice. And we will get to see that how they are doing it among the Indian patients. So when we started back in 2011, at that point, when my laboratory was starting, and in conversation with Dr. Kumar Prabhash, we kind of like realized that even was approved here in India as well. We did not know what was the one of the striking I missed to mention in the earlier slide is that these EGFR mutations, they vary by ethnicity. It is known to be about like 10-15% in the Caucasian population and about 25% and higher in station than in the Japanese population. So now, what does that mean? Is that the therapeutic compound, that is the EGFR inhibitor, can be given to only a small 10 to 15% in Caucasian, about 25% in East Asian. What was the incidence of EGFR mutations in the Indian population? And so we did not know how many patients would actually be benefiting with this inhibitor. So this was the first study that was performed, in which we were characterizing and we were profiling for EGFR mutations in 907 odd patients. And we were the first ones to establish that the incidence of EGFR in the Indian population was somewhere between the Caucasian and the East Asian. That was somewhere around 23%. Since then, it has one of the very highly cited publication where it has been very well established that EGFR mutation in the Indian population is somewhere around 23%. It's not just that these patients have EGFR mutations when they receive the EGFR inhibitors. Kumar's lab further went on to show that these patients, they do survive for even longer. And then we went on from there, from the EGFR mutation, to characterize the KRAS mutation in the Indian patients as well. And what we were finding was something very interesting that we were trying. We were getting to understand at that point of time in around 2013 and 14, is that even though these mutations, these major mutations, appear to be similar, these harboring even in the mutations, even in the Indian patients, albeit they were occurring at a very different frequency, as well. EGFR was occurring at 23%, KRAS was occurring at a completely different percent than what we kind of like get to see in the Caucasian population. In fact, they were showing a reciprocal thing. So, this work kind of like really leads to emphasizing that the EGFR treatment that were kind of like being offered to the patients, that how many patients could actually be benefited from there. So, when we move further, we wanted to go for beyond EGFR and KRAS. So there comes a computational student in my laboratory, my first graduate student, Pratik Chandrani, who is now a scientist at Tata Memorial. And Pratik Chandrani took a global approach, and he looked at several different therapeutically relevant alterations in lung adenocarcinoma genome. And what he found was Indian-specific mutations. What he found was mutation in another enzyme similar to the EGFR, but which was not characterized, or which was not found in this subtype, that is the adenocarcinomas of lung cancer, in the Caucasian population. Pratik went on to show that these mutations, that what he had found, what is shown over here is a heat map representation. Each column is a patient and each row is a mutation. And what he finds are significant mutations in FGFR3. What Pratik went on to show that these mutations, which are not there in the Caucasian population, and that is found in the Indian patients, were actually could be inhibited by a small molecule inhibitor similar to the EGFR. So here, what we show is the first landscape of mutations therapeutically relevant. That's what the clinicians would want to see. I mean, that's how the clinicians treat the lung cancer patient as of today. A patient harboring EGFR mutation, patient harboring FGFR3 mutations, or whatever. And so this was one of the major contribution that came out from the, and that was found to be as high as 5% of lung adenocarcinoma patient harboring FGFR3 mutation. And we went on to characterize these mutations. And why it was interesting is also because it was already known in head and neck cancer that another in the family of FGFR, that FGFR2, then it was mutated, and that leads to tumors in head and neck cancer. Then they were given an FGFR inhibitor. So this is a small pill that is given orally, and there is no infusions involved. And when you give an oral pill to this patient, in just 2 weeks, you could see this dramatic response. So the hope was that if that could be similarly be done to the lung cancer. But as a basic research laboratory, you would want to first establish that these mutations that you are finding in lung cancer patients, are they really activating? Are they really associated with cancer? So Pratik takes a whole lot of like approach in the laboratory in collaboration with other students. And what Pratik finds all the way to the he shows up to the mice is that these mutations are indeed activating. They cause tumors in mice. And when you give FGFR inhibitor, which are commercially available, such as pazopanib, we did not use pazopanib per se, but such as pazopanib, and you could see that there was a reduction in the tumor in the mice, suggesting that this could be one of a possible treatment in 5% of lung adenocarcinoma patients. Now, 5% of lung adenocarcinoma patients in the Indian population, given the huge denominator, is immense, and that may have a huge implication. So that was a finding from the basic laboratory. We will talk with Dr. Kumar Prabhash later on that how this can be taken up onto the clinical side. While in the meanwhile, I would kind of like give you a glimpse that is one of the major shortcomings that what we get to see in these targeted therapy or in personalized therapy. As I showed earlier, that there is a response that you get to see in just perhaps a matter of 2 weeks. But very often what is seen is that these patients, very often, they kind of like come back with the resistance that what they develop, some kind of a recurrence. And this resistance could be either an intrinsic resistance that's such that they would not respond to the treatment right from the beginning, or there could be something that they may have acquired over the phase of the treatment. So, so we wanted to understand that what exactly is this resistance in lung cancer, so that even before the resistance a patient kind of like develops, we would be ready with a therapeutic regimen that we can deal around with the patient. So another graduate student in the laboratory, Asim Joshi, he took another project again in collaboration with Dr. Kumar Prabhash. He took different samples which were longitudinally collected from patients, from lung cancer patients who were mutation lives, and at the same time when the rebiopsy was done, those who were before the treatment and then after the treatment when the tumor had relapsed. So Asim went on to develop also some lot of functional approaches, which I'm not elaborating as of today, by developing some cell lines which are already resistant, that what he is kind of like developed. So I'm not taking into the resistance aspect as of today, but I just wanted to give a wanted to let you know that we have performed a huge amount of work in the resistance. We have done lots and lots of sequencing of these genomes from the primary and as well as from the resistant tumors. And but that would be present. The story is still unpublished, and I would be happy to talk about it in some other session. What are the major leads from the resistance that what we have kind of like that we have got to understand based on Asim Joshi's work. Asim Joshi, another aspect that he wanted to look at, as what I said earlier, it was Pratik Chandrani and Asim Joshi both working on adenocarcinoma. We also been working on the another subtype, that is the lung squamous. This is again one of the first work. I mean, whatever I'm kind of like showing over here, these are all landmark or work from an Indian population. None of these work has been done before, and never ever before at this resolution we have been able to understand the complexities that what it kind of like underlie. So these studies have really allowed us to understand what is so unique about these cancer, and what are the possible therapeutic aspects that can be taken. So here is another collaboration, a huge collaboration work done. Again, I mean, but that cannot be much less emphasized that these work would not be possible unless you are working very closely with the clinician and in a center where both clinicians and basic researchers talk on a regular basis. So here is a lesser common lung cancer. There is a lung squamous cancer, and here is a 435 odd sample that Asim retrieves. He obtains these samples and he performs using a genomic analysis such as the next generation sequencing platform. And here is again a heat map representation of the mutations across different patients. Again, I'm not taking you into the detail. As what I tell you that the idea is to give you the glimpse of the discoveries that what we are making, and leaving you at the point that how this could be translated into the clinics. So Pratik, Asim went on to characterize. We have found some interesting candidates in lung squamous as well that has a potential of like being translated into the clinics that we will be working on to the next phase. Moving on, I'm changing gears now from that was the introduction. That was a brief overview of like what we were doing on the lung cancer aspect. And now I'm kind of like moving on into the using a similar approaches and using into into soft tissue rhabdomyosarcomas. This is a soft tissue cancer, and here again, here we were working with Dr. Bharat Rekhi from Tata Memorial, and this is again one of the first studies where one of the very rare cancer. It's very difficult to get like such huge number. This is one of the first study out there in the world to have a 49 set of samples altogether of rhabdomyosarcomas in just one study, and we could profile for a mutation in the transcription factor MyoD1, and based on that we could find that certain histological subtypes such as the sclerosing and the spindle cell rhabdomyosarcomas, they both harbor mutations in the MyoD1, and those patients which are harboring mutations in MyoD1, they were found to be having a far more aggressive tumor associated with it. What this study kind of like really revealed is that you really don't have to go for the histological characterization of these tissue. If this patient is harboring a MyoD1, it is going to be an aggressive disease, and that could be treated upfront in an aggressive way. So changing gears again and moving on, and now this time I'm going to talk about that. As what I said, that all the major cancers, the lung cancer, head and neck cancer, or the oral cancer, that what we're talking about here is one of the most prevalent among men in the Indian subcontinent. So another graduate student, this was primarily read by Pavan Upadhyay, and now more recently joined by Bhaskar Dharawat. And what Pavan Upadhyay kind of like did again here, he took different oral tumors. Again, this is one of the first study from the early stage tongue cancer. The ICGC effort or other different efforts which India has already taken has been restricted to the buccal mucosa cancer. In the early stage, this is the first cancer, and with a higher resolution details than what we kind of get the different genetic alterations. What Pavan finds is that all the mutations in the tongue cancer that you would expect to find based on the Caucasian population studies were consistent in the Indian population. But at the same time, Pavan made a spectacular discovery. Again, these are all Indian-specific discoveries. What he finds is that there is a mutation in Notch1. Now, I'm cutting a long story short. There are a lot of conferences about Notch1 being a tumor suppressor or a oncogene. Pavan did a humongous amount of functional characterization, and he landed up to show that Notch1 mutation, that what he finds in tongue cancer was amplified, and at the same time they were oncogenes, or they had an oncogenic role. Pavan went on to show, along with Dr. Sudhir Nair, a surgeon friend of ours at Tata Memorial at ACTREC, and together we showed that this Notch1, which was mutated, was found to be significantly associated. Very interesting finding of Pavan and along with Dr. Sudhir Nair. What we found is that this was associated with lymph node metastasis. So a tongue cancer which is having a lymph node metastasis, and if a patient happens to be a tobacco chewer, very likely they are going to be harboring a Notch1 mutation. So Notch1 inhibitors are already in clinical trials, so that can be given to these guys. So a patient walking into the clinic who is having a nodal metastasis is a chewer, likely to have a Notch1 mutation is higher. So now again, what happens is like we need to validate these findings by doing a series of clinical trials. We will talk about it in the next phase. So what we are doing now is that now we are kind of we have gone more sophisticated in our characterization. We have now made mouse models. So here, a postdoc, a very talented postdoc in the lab who has joined Dr. Ashwin Butle. And what he's shown is that what we are doing is an orthotopic tongue cancer model development. So what it does is that, so here what you're seeing is an anesthetized mice, which has been anesthetized for the transient period. And here is what you're seeing is an injection. It requires a lot of dexterity, very tactful injection of tumor cells right into the tumor right into the tongue of the mice, and to have an orthotopic oral tumors. And we have been very successful now in doing that. And what kind of like shows that these signs injected, you could see that these tumors kind of like start forming up within a matter of like about 30 days or 35 days. And so now we have an orthotopic mouse model system up in the laboratory. We can ask questions which otherwise would not have been possible. But then again, we have to bring it to the clinics along with the clinical friend of ours, and to see that whatever hypothesis that what we built to test in these guys could actually be relevant from the human testing as well. Changing gears, we talked about lung cancer, we talked about rhabdomyosarcomas, we talked about lung cancer. Now I'm talking about another Indian specific disease, gallbladder cancer, which is considered to be overall across the globe. It will be called as one of the rare kind of like cancer. It is not found that common except for a few countries such as South America. In India, gallbladder cancer is very much common in the Indian Gangetic Plains. Up there, the cancer incidence is very high. Another graduate student, Prashish Iyer, joins the lab, and he was interested in gallbladder cancer. Again, he takes a similar approach. In collaboration with Dr. Shailesh Srikhande, we get all the gallbladder tumors and high quality gallbladder tumors, and he performs next generation sequencing platform. He uses next generation sequencing platform to characterize these genomes. And again, cutting a long story short, Prashish made a fantastic finding of mutation in ORB2, HER2. Now, why is this fantastic? Because this is another enzyme which is already for which the treatment is already there in other cancers such as breast cancer or even colorectal cancer. And Prashish finds ORB2 to be mutated, the same gene to be mutated also in gallbladder cancer. A lot amount of work Prashish had done, a lot of like functional characterization to reach to a conclusion that 40% of these gallbladder cancer are likely to be harboring in this gene. So 40% of these patients are likely to benefit from this treatment. And Prashish again goes on to show on using a mice model, the xenograft assays that these mutations which were derived from the patients, Indian gallbladder cancer patients, when they are brought onto the mice, they form tumors, and when you give them afatinib orally, which is an ORB2 inhibitor, you find that the tumor kind of like regresses, which is a very strong statement that and afatinib is already approved for other diseases. That can be adopted for treatment for gallbladder cancer. And mind you, there is no treatment of gallbladder cancer in targeted therapy as of date. So this is a very strong candidate that what Prashish had found. In fact, Prashish analysis goes far more sophisticated and far more detailed that I would be able to talk about today over here. But I would just want to emphasize on that Prashish found that there is a co-occurring mutations in gallbladder cancer. Along with the ORB2, he also finds mutation in KRAS. And he finds that only those patients which are having a wild type KRAS, similar to what is like a paradigm and the algorithm that you follow in colorectal cancer, need to be followed in gallbladder cancer. If there is a wild type KRAS along with an ORB2 alteration, give them ORB2 inhibitors. However, if there is a mutant KRAS, these are not going to respond. And this is exactly what he kind of like went ahead to show even in the mice. Another graduate student, so I'm moving to the last cancer type. Another graduate student, Trupti Togar, she has just submitted her thesis right now. This work was performed with Dr. Supriya Chopra here at Tata Memorial at ACTREC. And this is again one of the first studies or such a comprehensive studies with about 99 cervical cancer patients. One of the most comprehensive work in cervical cancer from the Indian subcontinent with both the subtypes, huge amount of lung adenocarcinoma, which is a subtype of cervical cancer, and Trupti goes on to use data from the cervical squamous as well, which was earlier kind of like published. So I wouldn't take you into the details, but again, Trupti's work has really brought us to a very exciting crossroads that we want to excite our clinician friends. Is that this is again with the mouse model. She finds that for the gallbladder can also be extended to a small sector of cervical cancer. In fact, we have found other therapeutically relevant alterations as well in cervical cancer, but I'm not talking about like that today. This is statement using mice as a model system, xenograft assays. Ashwin is now developing orthotopic cervical cancer model. So I mean, we would be able to validate this far more across. So, but the question comes up that what are you really going to do with these kind of like alterations if a basic researcher perspective? I mean, you find these alterations, you publish, most of these work get published in high impact journals, peer-reviewed journals, international peer-reviewed journals, but then so bringing it onto the clinics, therapeutics takes a lot of like efforts and time. However, in the meanwhile, we have developed another tool which can be immediately implemented to the clinics, and what is kind of like is currently on the way. So this was again the first graduate student who is now a scientist at Tata Memorial, Dr. Pratik Chandrani. He is working in the group of Dr. Kumar Prabhash now and the medical oncology department. He's an independent scientist. He developed another tool that is a Clinome. Clinome is an automated computational tool that allows clinicians to take raw data from a next generation sequencing platform. And you know, when you're in the clinics, you don't want to get bogged down with the details and the complexities of the noises and the nuances of the next generation sequencing platform. All that you want to know is that based on this data that has been generated, if there is a relevance, a therapeutically relevant target or not that the patient can be given. So this is what the tool kind of like does. It goes into the details that I would kind of like this cartoon I would kind of like skip for now. It takes a lot of filtering and statistical rigor that is all built in, which is an automated tool. And what essentially comes out is for a clinician is that it comes out in a format of automated report. It tells you only the relevant information in a very pragmatic manner what the clinician really wants. So here is a patient and it tells that if this patient is harboring this mutation in an enzyme, there is an EGFR, and if this is harboring, then if there is an US FDA approved compound, which is already approved by the US FDA, these are all automated report. It just populates over here. Here in this case, it tells you that there is a treatment, gefitinib, but it is a treatment in a lung cancer, not for endometrial cancer. So that's why it populates in a section called as off-label. And it goes into the much more of a sophistication. It also puts out all the clinical trials that is being done. And it populates and gives a link over here. So if the patient was really wanting to, can go and enroll himself or herself to this clinical trial. It also tells you that what are the compounds which have been tested that because of the other co-occurring mutations would not be effective in this patient, because this patient might be very likely be resistant to those. So this is the kind of like automated report that kind of like generates that Tata Memorial has tested for quite some significant time, and we have data now from the patients that this report kind of has really benefited the patients. These are the different details that comes out from that report that it kind of like generates in an automated manner, and but I wouldn't go into the details of it, and this work is yet to be kind of published. So this Clinome does a lot many more things. It also kind of like excites basic researchers besides the clinicians. It also tells what are the other different kind of like incidences and what are the incidences of those mutations across different tumor type. But I'm again not going into the detail of it. All I say is that it is a very robust tool that has been generated and ready to be translated into the clinics, and which has already kind of like been done so at the Tata Memorial. It has been tested against more than 40,000 odd samples that Pratik has already done at ACTREC. So finally, what we have kind of like talked about from the basic component is that we started with the characterization of the lung cancer. What I missed to mention is, along with Dr. Kumar Prabhash group, when we characterized the 907 samples, that also allowed us to identify, also allowed us to develop a technology, a methodology to test these mutations in the clinics, and this allowed us to bring down the cost from $250, that was in 2010 or earlier, to $12. approximately, and that was already being rolled out at Tata Memorial. However, now Tata Memorial has also moved on from the conventional technology. Now Tata Memorial has moved on to the next generation sequencing platform. So we have talked about the findings in adenocarcinomas. I just mentioned about what has been translated into the clinics. We talked about the ASIM's work on lung squamous. We talked about briefly about the work done by Dr. Bharat Rekhi in rhabdomyosarcoma. I just gave you a touching thing about the Notch1 mutations in head and neck cancer and the orthotopic models and all that in the tongue cancer that we have developed. I did not talk about the MMP10 or the work of like Bhaskar Dharawat as of now. We talked about the gallbladder cancer in Shailesh Srikanth, and of the colorectal cancer can be adopted to treating gallbladder cancer. I mentioned briefly about an overview of the findings from the OB2 alterations, that is treatment with afatinib in cervical cancer. This work was done in with Dr. Supriya Chopra, and just kind of like gave an introduction to Clinome. That has already been translated and that needs to be further developed to take it more beyond. So with this, I would bring an end. What I've not talked about is breast cancer and the huge initiative that we have done along with Dr. Sudeep Gupta and Dr. Rajendra Bhatve, and several other different kind of like initiatives that has been happening in the lab, and other arms such as the computational arms and things that what is being developed. I've not kind of like talked about. So whatever work that has been done couldn't have been possible without a huge effort of my group. Here are the team members who work in the functional laboratory. Here are the computational students who have been working around, and here are the collaborators at Tata Memorial with the different tumor types that we have been working. These are the people who are kind of like graduated, kind of like moved on from the lab, without whose contribution this wouldn't have been possible. So, so with this, I would kind of like stop. Not exactly sure if I have exceeded my time, and then I would back to Hitesh, and then back to the intrinsic part for Dr. Kumar Prabhash. Thank you.
Thanks, Dr. Dutt, for such an insightful talk. And it's very interesting to see how different biomarkers are identified in Indian population, right? And thanks for talking about your research in different cancer types. And I think now it will be very interesting to hear from Dr. Kumar Prabhash on how this research translates into clinic. And thanks, Dr. Kumar Prabhash, over to you.
Good evening, everyone. And as you heard a wonderful talk about how important it is to have the molecular data from our population, and also how it changes the way we start behaving and treating once you have the data. So what I'm trying to do is, I'll use lung cancer, because that is one of the cancer I treat, and which me and Dr. Amit, we work very closely as a prototype to how it has made a difference in our patients. And also I'll give some example how worldwide also it has made a difference that we have those molecular data available, and the clinician and the basic scientists, they're working together. And here you see, I'm not presenting alone. This whole work is by a group, and you see all the names there. Starting with the first slide, and this slide becomes very important to understand how much difference we have made as far as the treatment is concerned based on the molecular data being available. Not long back, this is in 2002, when work of having best of the treatment used to give average life of the patient in advanced lung cancer of around 8 months. And that was considered state-of-art treatment at that time, and this was the NEGM publication. So this number of around 8 months will be very important to remember. And remember, this is not the whole patient, this is a selected patient for the trial, and that may give you a hang of it, how the newer treatment and the technology has made a difference in the outcome. So as I mentioned initially, starting my presentation, that molecular profile becomes important. And this was a bit discussed by Dr. Amit. And this, you will keep hearing this many times, Dr. Amit's name and his group. This was the work where molecular profiling of lung was there. And also there was a discovery. as a clinician, this curve was very important, and Amit mentioned it. And we picked up from the curve the various mutations which we see in Indian population. And this was the first time when we had the fair extensive molecular profiling. And as compared to Western population, we see the certain differences. And this is what we took forward. And what we took forward was to have this test in the clinic and have this test in the clinic at the cost which is available to almost everyone. Meaning that almost every patient coming to our hospital was able to get this test done as far as the EGFR mutation is concerned, because treatment was available, which was again useful and available and cost effective in almost every patient in our clinic. This study also showed that if you treat the patient with the targeted therapy, when you have a mutation, then they do better as compared to if you don't have a mutation, and yes, in that situation, you are not able to treat them with a targeted therapy. Because we had this test, and this is important to realize, because molecular profiling was done in house in a collaboration. It was made cost effective. So every patient of non-small cell lung cancer who requires the mutation to be done, especially EGFR mutation at that time, so we were able to do in every patient. This led to not only validating this that the test is good and patients do differently if you have a mutation or not. This also led to this randomized trial, and this was the first randomized trial in lung cancer with EGFR mutation in our country, where we compared that if you do have a mutation in advanced stage patient of non-small cell lung cancer with EGFR mutation, comparing them with molecular therapy, that is targeted therapy. Here it was oral tablet, gefitinib, compared to chemotherapy. What it showed that if you are able to give the patients who have got EGFR mutation, positive patients, then the disease control is far better as compared to if we give them chemotherapy. And what we see here, the value of hazard ratio of 1.5 is similar what we see in Eastern Asian population or in the Western world, suggesting that our patients, if we give them the treatment of EGFR targeted therapy, they do as good as what we see in Eastern Asian population or what we see in Western population. The lower curve shows the survival data. Remember what we started with, that was around 8 months of median survival. And what it shows here, that if you have EGFR mutation positive patient and you are able to give them targeted therapy, then the average life of the patient becomes around 20 months, and that too with a tablet which is easily affordable and that is fairly tolerable. It is a bit like giving treatment for hypertension and diabetes, where the people take tablet at home with some side effects which can be managed. There was some other work which developed subsequently to this because of this collaboration. This was a multiple collaboration. Again, Dr. Dutt being pivotal to it. We had some samples of the patient which had a family history. We took those samples and the whole exome sequencing was done. And what we found was there were some mutations which is not so well described at that moment, that was in HER2. And because this result was available, we were able to give these patients those treatment. This was only 3 patients, but it highlights that in many situations, and we were able to give the therapy, and you can see the responses for these patients. Because this test was available, highlighting that many times you can have information for individual patients which can be useful in giving the therapy to these patients. These patients who would not have survived beyond 2 or 3 months, they did survive beyond few years because this test was available, and based on test, we were able to give therapy which worked on HER2. So one of them we gave trastuzumab, and another one we were able to give lapatinib. Again, because these works were going on, there were many other things which came out, and one of the things again was in collaboration with Dr. Dutt, was we had multiple patients which we had more than one mutation, because data was bigger. So we did have these 3 patients, and where till recently when we had analyzed this data, we were not sure about it, that if you do have a KRAS mutation and if you do have a EGFR mutation, how they are going to behave. Because the treatment data was available, we could analyze the data that at least in our population, if you have EGFR mutation and if you have a KRAS mutation, then KRAS mutation doesn't override EGFR mutation. If you are giving them TKI therapy, that is EGFR TKI based therapy, they respond. But this data was available only because we had the data from the lab that you do have these double mutation available to these patients. Otherwise, this data would not have come in public domain, and that was primarily because there was collaboration between the clinician and the scientist in the lab, where the exchange of information was there. And you can see the the person who was working in the lab with Dr. Amit Dutt, student photograph down on the left side. Taking it further, we were doing this mutation analysis in many tumors, and we were also did this mutations, especially EGFR mutation in squamous cell cancer. Data has been pretty scanty, but because every patient coming to Tata Hospital got in non-small cell lung cancer patients had EGFR mutation testing, so we could analyze the data for EGFR TKI in squamous cell cancer. And what we see here that in squamous cell cancer also, if you have a EGFR mutation positive patient, and if you give them targeted therapy, that is oral tablet, they do better than if chemotherapy was given, highlighting again the importance of working together and exchange of information between the lab and the clinic. And this happened only because we had the information from the lab to the clinic. As again highlighting that we were doing a lot many mutation for a lot many tests for patients coming to our hospital, highlighting some of those numbers. What you can remember is we register almost 2,000 odd non-small cell lung cancer patients every year at Tata Hospital, and almost all who require this testing, it gets done. It allowed us to analyze the data regarding those relatively uncommon EGFR mutations. So we know that there are common EGFR mutations on exon 19 and 21, but you do have various combinations of uncommon mutation in exon 18, 19, 20, and 21. Because this data was available in 80 plus patients, again, this is one of the largest series in this situation, we did learn in collaboration with the lab, again, that many of these mutations don't behave similarly. And what we see that in uncommon mutations, you have a median survival which varies from 2 months to almost 28 months, highlighting that we need to learn more in this situation as far as the uncommon mutations are concerned. And many of them, they don't behave similarly. And as the time goes by, as the number increases in these individual segments, we will learn that these exon uncommon EGFR mutations in lung cancer will have a different kind of therapy. We also analyzed the data for exon 20, and again, it taught us in exon 20 also different subtypes may have a different outcome. And this has gives us some perspective that in this situation, in those uncommon mutations, we need to subdivide this data, and then maybe we can have a strategy for how we are going to manage these patients. At this moment, we club all those uncommon mutations together, and we are trying to do it because now we have the database of almost 6,000 plus patients, and we'll try to see that how we can divide these uncommon mutations in different subgroups so that we can develop those effective treatment in this setting. We had a bit of a saturation for the treatment being available for EGFR mutation positive patients with those TKI. That was the first generation TKI. And globally, people were trying to develop the further treatment. So people have developed other tablets that we call it second generation TKI, or we call third generation TKI. This is the data regarding third generation TKI, and what we can see here, they compared the third generation TKI as compared to the first generation TKI in EGFR mutation positive patient. What it shows that yes, the newer drug, that is third generation TKI, it adds life to the patient. So overall survival improves in these patients as compared to giving first generation TKI. But what I want to highlight from Indian perspective, that this drug is not available for almost 9. It is available in the market, but not available for almost 98% of the patients. So one can take it, absolutely fine, please take it. But we know that for majority of the patients, we required some other treatment which can improve outcome. And again, because we had lab data being available, so we tried to develop something which is useful for our patients. So we thought, can we have a similar outcome, what we have seen with the newer drug, which most of our patients can't take it, so that we have a better outcome in our patients. So what we did was we compared those targeted therapy, that is first generation TKI, to first generation TKI plus chemotherapy in these EGFR mutation positive patients. And because what we found that this combination is definitely affordable. Almost every patient coming to Tata Hospital can get this combination. We did this randomized trial in 350 odd patients, and that is one of the biggest number in this setting. And in this setting, it may be one of the largest study. And what we found that if we give this combination, and as compared to the first generation TKI, the disease control doubles, and the overall survival improvement at around 18 months is almost 20%. And it looks similar to what you can give the patient the treatment and the outcome what we have with the newer drug, that is osimertinib in this setting, but at the fraction of the cost. And the cost will be almost 1/15 or 1/20 of the cost what we have with osimertinib for our patient. But I need to highlight, this happened only because we had the support from the lab and the collaboration with Dr. Amit and his team and our diagnostic lab at TMH. And just highlighting how was the impact of this work. Sometimes we want to highlight that it did impact, and it had some decent impact. Otherwise, also this was in the ASCO oral presentation in the Daily News. It was the top 30 abstracts in the ASCO. It was now already a part of standard treatment. If you see this guideline, you can see that this is a part of the guideline for treatment in as a standard of care for patient having EGFR mutation in lung cancer. This is also as a part of ESMO guideline, and just highlighting here that combining this chemotherapy with TKI is one of the options, and with the grade A recommendation suggesting that there's a good evidence for this drug to be used in the clinic. And this was a write-up in the Nature Review for the same study, where they also agreed that this is one of the standard of care for our patient, and there was some decent coverage across the board that this was an important study, and it has some decent impact. But for me, the most important impact was that with this treatment, which every patient coming to Tata Hospital, if they require it and if they are fit enough to take it, we can give it. Cost will not be an issue to give this treatment. So that was a satisfying moment. But again, highlighting the importance of the clinician and the lab working together. Now, how far we have moved, in EGFR mutation positive patients. Remember, we started with 8 months of median survival, and you have at least 4 randomized trials showing that average life of these patients has become now 40 to 50 months. So we don't talk about in 5-6 months. We talk about in 5-year survival here. Here around 3 to 4 year survival. Again here showing around 3-year survival. Again here showing around 5-year survival. So in lung cancer, which we talked about 6 to 8 months median survival, we are talking about in this subgroup, that is EGFR mutation positive patient, if we are able to give appropriate therapy, but this appropriate therapy is totally dependent upon the lab. Then we can have 40 to 50 months survival and 5-year survival of around 40% in these populations. This also has developed only because not only we had this lab report or lab information at the beginning. This also has happened because we had the information from the lab, what happens later on, and which also helps in deciding the treatment and which leads to this substantial improvement in outcome. Now, what we see, we started with a treatment option of only 1 treatment option in this segment, and now you have almost 8 treatment options in this segment. suggesting that lab feeding the clinic and the clinic feeding the lab can lead to substantial improvement in outcome and substantial number of options. So now we have lot many options, which becomes tough to choose, but it is a good situation to be in. We also had an issue regarding, we had almost 35% of the patients which we had to start treatment because the reports were not available. And this we had a constant discussion with our lab. And again here, the diagnostic lab and Amit Dutt and its group, they came together. And these are the two work, and this is one of them, where we try to see what is the concordance of liquid biopsy to the tissue biopsy. And what we see here, that's around 80% odd. This is another work showing that overall concordance of 90% plus. We know that sensitivity is low, it is around 90%. We do miss around 10% of those EGFR mutation positive patients, but it does highlight that in case, if you don't have a tissue biopsy, in majority of the patients, you will have this report based on which you can give treatment, and that becomes a very useful solution to our patients. This is another work which we are trying to do and see that can we have those sequential liquid biopsy data for patient on treatment, and we are trying to figure out how we can integrate. At this moment, we can only say that we are learning to understand how we are going to integrate the data of liquid biopsy at different time points for patients on EGFR TKI therapy. Do we have this only one example, or we do have another examples where we know that if you have the very important lab information report available, then the treatment outcome changes. This is the ALK positive non-small cell lung cancer data from TMH, and these were where we have treated the patient with TKI if we had the ALK positive report. And what we can see here, that 5-year survival was 33%. Remember, average life of the patient of non-small cell lung cancer in advanced stage used to be around 6 to 8 months, and then here we are talking about 5-year survival of 33%. And if we give crizotinib, then the PFS increases or survival increases further, suggesting that again, that is not one-off. If you have ALK positive patient, also patient does better. This is the international data suggesting the same, that if you have ALK positive patient, then the 5-year survival is around 40% or plus in these patients, emphasizing the importance of what we get the information from the lab. Now we have multiple treatment available in these patients. These are some of the names. What we are slowly realizing that what are the mechanisms of resistance, and this is another work which Dr. Amit Dutt and we are doing together. Data is preliminary, but we hope that these information of resistance mechanism for EGFR and ALK, which Dr. Amit Dutt's lab are integrally involved and they are focused on, will give us information which may help us develop treatment for our patients. Now, do we have some other examples? And I'll go continue with some of the example in lung cancer. We do have a ROS1 mutation in non-small cell lung cancer. This is again our data for ROS1 positive patients, which we analyzed recently, and showing that if you have ROS1 positive patients and if you give a TKI, then they survive better as compared to you are not able to give TKI in these patients. And international data also suggests in this situation we talk about again 4-year survival, not the average survival of 6 to 8 months, highlighting again the lab data feeding clinic becomes very important in outcome of these patients. And as the time goes by, slowly we are learning that ROS mutation also you do have different drugs, different mutations which develops as the time goes by, and again the clinic data feeding the lab to develop the better drug in this situation. Do we have some other example? Yes, if you have a BRAF mutation and you are able to give targeted therapy single agent, then the response rate is around 30 to 40%. If you're able to give 2 drugs combination, the response rate goes to around 66% or 67%, highlighting again that importance of doing the testing and importance of treating based on the test being available. And this was mentioned by Dr. Amit Dutt that about NGS, and this is where it becomes important. Think about, we talked about by now 4 mutations. I'll be going subsequently also some other mutations, that how do we have all these data available upfront? If we do sequentially, we won't have adequate tissue. and it will be cumbersome. Repeat biopsy is almost very unlikely in our patients, and globally also this is the same information. So NGS becomes important, where you can have all this information available upfront for these patients. Again, developing those NGS in cost-effective manner becomes important. This is another data showing that if you have met mutation and if you give tablet to the patient, almost 4 out of 10 patients they respond. If you have a HER2 mutation, then almost again 4 out of or 5 out of 10 patients they respond to the treatment. Something interesting, what we call TRK inhibitor. If you have anti-RK test being positive and you are able to give them anti-RK inhibitor, then 9 out of 10 patients or 8 out of 10 patients they respond to this treatment. What this highlights is that you have at least 8 or 9 molecular changes for which you have targeted therapy available, and they are part of the standard therapy in NCCN guidelines. Now, if we want to have these reports available upfront, we need to have a technology, and the technology should give us all the reports, and also it should be cost effective and useful. And this is where again the interaction and the feeding of the information from the lab to the clinic and the clinic to the lab allows the development of this treatment and also development of the test which can be available to majority of our patients. This is another work of ours suggesting that in the patient who are bedridden, which we used to send them home without any treatment, if you can give them tablets and if they are EGFR mutation, then they also live a decent life for a year or beyond in this setting. This is highlighting that none of this would have been possible without having all these names, or what you can see here of the all the students from Dr. Amit Dutt's lab and the top, and you can see below all the coordinators and the residents from the TMH, which works in medical oncology department, and Dr. Amit Dutt's lab in this setting. And these are the key people. What you see here, all the individuals who are the consultants in the lab, in the clinic, which ensures that this work happens. And this is the diagnostic lab team, which is there and which functions, which ensures this collaboration is steady and strong. And these are the key people who allows this research to happen. You require those mentors who when you land up in hot water, they ensure they take you out of the hot water and ensure that it happens. And first one is my guide, Dr. P.P. Babsey. Second one, Dr. S.T. Banavli, my ex-HOD and now the academic director, very supportive. And the below one, who is the TMC director, and now also he remains the TMC director, very supportive. And that allows for these kind of works to happen and move forward. And these are the acknowledgement to various people who have worked together to ensure that this happens. And this is my family old photograph, but yes, those small ones, and my wife, and sure it takes a lot of my time from the family to get these done. This is very important, and thank you so much.
Thank you so much, Dr. Kumar, for such an elaborate talk and such an informative talk. I'm sure that patients would be very encouraged to understand how clinicians and researchers are working together to identify new ways of treatment which would have better clinical outcomes. So I think it's very exciting. I think we have time just for a few questions, but one thing that I wanted to ask both of you. So what is one thing that you think is the secret to your successful collaboration from research perspective and from clinical perspective?
Asking me or Amit?
Both, one from research perspective, one from clinical perspective. What do you think has worked for both of you to build such a successful collaboration?
Yeah, Kumar, you can go ahead.
Okay. So, see, the most important was on the both sides, willingness and the passion. So, Amit is a very passionate researcher, and another thing was we used to ensure that we meet every week.
Right.
Except for last few months because of whatever COVID and some other things, it has hardly been ever that we have not met on Friday. So it was like we had a fixed date, on Friday, and I used to ensure that he gives me a tea. But that
Right.
tea used to have a whole lot of discussion. That is to ensure that, and that also had a shared accountability. So that
Right.
small comments with each other, that what is pending, you remember it, and then you again, because you know that you're going to meet next week, you exchange ideas, but you meet, and then there is a not only we used to have tea, the discussion used to go all around, and there was some passive discussion about what has not been done, which we had discussed last time. So there was a passive accountability to ensure that when I meet next time, we meet each other, we have done part of it, the work. And third and important thing was absolute transparency and honesty. So we were very clear about it, what we are able to do and what we are not, because there are challenges everywhere. So if I'm
Right.
not able to do certain things, we used to be very open and say, Amit, this is where we are getting stuck. But what it led to some solutions. So if not able to do something in one area, we used to go to the second area. And because there's so many unmet needs for our patients. There's so
Right.
many unmet needs. So we were able to move forward.
And how do you think industry can play a part in such collaborations and helping out?
So it is something. In fact, we had this discussion so many times because Amit is trained in US. I have visited and I have spent some time there. But Amit is trained, and he has been trained in one of the best of the lab in US. So it is something which is usually lacking in our country, that somehow we are very suspicious about each other, the academia to to the industry and industry to academia, and that is one of the important reason why you may have something good, but to bring it to the patient becomes very tough, because that is somehow academic centers can't do it. It is impossible to do that. So uh, I don't have a prototype to it, but I think so. Unless we try, unless we fail 5 times, we won't find a prototype.
Right.
So we
Right.
need to try and find out. And we have kept trying. I don't say that we have succeeded in a big way, but industry, commercial organization should be integral part of research, in our country, if you want the test and the treatment to be available to majority of our people.
Right. Thank you. Let's just take 2-3 questions from the audience. So one of the question is there from Dr. Rohan Shetty, which says that if there is a concomitant EGFR and ALK mutation, do you treat with both the drugs, sequential or concurrent?
So this is where I'll say no, lab work is here. I think so. I'll say easy, that you have a test. We don't know, really speaking. So we have almost now 18 or 19 patients with these two mutations. And
Right.
initially we were treating with EGFR TKI. Some patients we were treating with ALK TKI. Last two patients we have treated with both, because we found, is it safe or not, we try to see that. We found that it was okay giving, combining both the drugs. So I am not able to say today that how ideally to treat, but last two patients we have treated with a combined TKI. Previously it was easy, because what happened, you give EGFR TKI first and then give ALK TKI later on. But now in both the setting you have at least second and third line treatment. So how do you manage
Okay.
them? Because when they progress, which part has progressed, where the disease has increased, which mutation is driving, we don't know.
Right.
The
Right.
tricky situation. So at this moment, we are giving both the TKI.
Okay. And there are a lot of questions. Yes, Dr. Dutt, please. Dr. Dutt, you're on mute.
Amit, you have to unmute.
Yes. Sorry. So I would just want to add what Kumar was just mentioning. One of the important aspects that one need to also take into consideration is the allele fraction. So what it means is that you would want to know whether this ALK mutation or the EGFR mutation, they are allelic or not, or for that matter, in a simpler words, if they are clonal or not. What that means is, if it is the same cell which is harboring EGFR mutation and is also harboring the ALK mutation, it is very likely, given that solid cancer or solid tumors are heterogeneous in nature, that these could be two different clones. So if there are two different clones, so as what Kumar is mentioning, then having a combined treatment perhaps might be effective. However, if it is in the same clone, then perhaps even a single treatment might be equally effective, or we really do not know. We do not have enough of preclinical data to differentiate between or to discern between these two. But comparing with the allele fraction, you would be able to decide whether it is in the same clone, if it is clonal, or if it is not clonal.
And these kind of information are available in the.
Oh yes, so for example, if you're doing if this information is coming from an NGS platform, you'll always have an allele fraction associated with that mutation. So if you would compare the allele fraction of an EGFR mutation with an allele fraction of an ALK mutation, and if they are comparable, that means very likely they are from the same cell.
Right.
And if they are completely different, then it is very likely different clones which are harboring the mutations.
Right. I think one last question I'll take, because we've got a lot of questions on, I think, FGFR3, because everyone is very excited about this particular gene.
But before we get on to the FGFR3 and other questions, Hitesh, if I could just get back to the collaboration between a basic researcher and Kumar. Kumar has very elegantly narrated, and he has kind of like mentioned what exactly are the basic and important component. But I think this is something which is important, and that's why I want to bring it, even though we have gone ahead of it. One thing which is kind of like very important for anyone who would want to have these kind of like collaboration, I do agree that we have had a very successful collaboration with Kumar and myself here at Tata Memorial Center. One of the reasons is a mutual respect. Mutual respect, of course, Kumar has already mentioned about the passion. Both have to be kind of like genuinely interested, but then you have to also believe in them, because very likely you see a huge gap between a basic scientist and the clinician. So unless you are good friends at a personal level and you have a mutual regard for each other, a collaboration somehow I have not seen to kind of like go with. So most of our clinician consultants over here, they're very good. So a friendship. So that's why a cup of tea, as what Kumar was emphasizing, is very important. You may talk something which is not related to science, but you develop a friendship, you develop a mutual regard and a mutual trust for each other, and then things will all fall in line.
Fantastic. Thank you so much, Dr. Dutt, for this information. And the last question is on FGFR3. There are a lot of questions coming on that. So if someone finds FGFR3 mutation today, what is the best treatment option that could be available? Could it be used as a standard of care?
I will leave it to Kumar.
Can you repeat?
If FGFR3 mutation is identified, then what would be the best line of treatment? And could that be recommended as a standard of care as well?
So if you ask me, I maybe not in first line setting, but we know that FGFR mutation in bladder cancer, already there is a drug approved. So in second line and beyond, sure that FGFR inhibitor can be considered in this setting. We are hoping that drug comes into the Indian market soon. In absence, yes, there are drugs which are what we call dirty kinase inhibitor that can be used like pazopanib. And yes, if you ask me in subsequent, maybe not in first line setting, I won't say in first line, but second line and beyond, sure, that can be considered in these patients. Yes.
Sure. Fantastic. Thank you again so much for your time. And I think the takeaway message for everyone would be that for a successful clinical and academic collaboration, the most important part is the mutual respect for each other as collaborators, the passion for the work that you are doing, and you need to spend significant time together to make sure that something interesting comes out. I guess I have been able to sum up your thoughts on that. And would like to thank all the attendees also for the fantastic response. The video will be available very soon. We have another exciting talk on 22nd of August by Dr. Sharon Law from Singapore, and she'll be talking about challenges in pediatric glioblastoma. Please note, most of our lectures have been at 7 p.m. India time. This one would be a little earlier. It would be at 2 p.m. India time. So looking forward to welcoming you all. Thank you again, Dr. Kumar, Dr. Amit Dutt, for this fantastic talk. And we're looking forward to seeing much more collaborations like this from India and much more exciting work from your labs. Thank you again. Thanks a lot.